In this video, we will continue our discussion about this pancreatic cancer invasive characteristics. So, continue. We'll continue our uh, story from our previous videos. That in the previous videos, they have identified that it's the TGF beta uh, inhibitor that prevents the invasiveness of the pancreatic tumors and also prevents the ablation of the endothelial cells. So now they wanted to check uh, which signaling pathways actually promotes this invasive characteristics of the pancreatic cancer. So the reagent that they have used to inhibit this TGF beta actually act on three different pathways, that is ALK7, ALK5, ALK4. And however, when they try to do this CRISPR knockout of ALK4, they are not able to do the CRISPR knockout of ALK4. So they mentioned in the paper that ALK4 is important for this pancreatic or both the cell survivals. However, they did the study on ALK7 and ALK5 knocked out cells. So this is their in the panel A. This is their 2D experimentals timeline. How they did the 2D experimental timeline. So they create an annulus ring, and within this plate they grow the pancreatic cancer, and on the periphery they grow the endothelial cells. It can be seen here. So when this uh, uh, so when this annulus is been removed, so what will happen is that the tumors will starts to ablate the endothelial cells and spread across in diameter. So this indicates the invasiveness properties and also the ablations of these endothelial cells. And this is how they validated their 2D experiments, is that when they use this TGF beta inhibitors that they have used, the SB431542, they can see the decrease in the uh, invasiveness of these pancreatic tumors. At the same time, when they use, in the case of control, they do not see a decrease in the invasiveness of this invasiveness properties of these pancreatic cancers. And this is what and in the third experiment, this is what it is shown, and this gap has been filled. When this gap is being filled by these pancreatic tumors, then they consider it as time zero, and from there they uh, starts to observe the invasiveness of these pancreatic tumors. So next, what they did, they did individual knockout both in the case of uh, endothelial cells and also in the case of pancreatic tumors. So when they did it for the endothelial cells, they do not see any decrease in the results, and those results are there in the uh, supplementary documents. However, when they did the knockout of this ALK7 knockout in the pancreatic tumors, they can see the decrease in the invasiveness area. And however, for this ALK5, they do not see any decrease in the area, and those results are also in the supplementary figures. So from this they come to know that it's the ALK7 signaling pathway that helps in this invasiveness property of this pancreatic tumors. And thereafter they need to check the which ligands actually promote this ALK7 signaling pathway. So when they compare this mRNA expression of this normal pancreatic cells and the these pancreatic tumor cells, they can see the high upregulations of this inhibin beta alpha, beta A and inhibin beta B compared to the normal pancreas. So this indicates that the high presence of this inhibin beta A and beta B could be the triggering factor for this ALK signaling pathways. So they use the reagent that is follistatin that inhibits this inhibin beta A, and uh, when they check this invasive area with this follistatin treated cells, they can see that there is a decrease in the invasiveness area compared to the control case. So this also made their point that it's this uh, inhibin beta A that helps in triggering of this ALK7 signaling pathways in the pancreatic tumors. And they also then quantify this quantify these biometric blood vessels that are replaced with the pancreatic tumors in their 3D model. And in this case also they can see that when this polystatin is used, there is a decrease in the uh, area. There is a decrease in the pancreatic tumors area that has been replaced, uh, that replace these biometric blood vessels compared to the control case. And after validating the in vitro results, they do the similar study on the tumor cells, where they use the tumors with this ALK7 knocked out. And in this case they can see that the number of density of the endothelial cells is high, and also there is density of these apoptotic factors are low. So these things are also in line with their observations from these in vitro studies. And thereafter and this is the we can say immunofluorescence image validating the quantitative results, that compared to the this scramble case or control case, they can see the more endothelial cells or red signals in this ALK7 knocked out. And this is the individual images. And in this case also we can see that the apoptotic factor or caspase signals are high in the control case compared to the normal case. And when they check these apoptotic factors in this endothelial in vitro studies, they also see that when this ALK7 is used, the apoptotic factors are low and are low. However, when they use this uh, when they use the when they did the control experiment, they can see the apoptotic factors are high and are at the interface of this pancreatic tumors and the endothelial cells. So all these results are in line, and it indicates that it's the ALK7 signaling pathways in the pancreatic tumor cells that promotes these invasive properties of this promotes these pancreatic tumors invasive properties, and helps them to migrate to this another blood vessels and ablate the endothelial cells. And these studies are also been done on two other pancreatic tumor cells, that is PANC1 and BXBP1. If you need to understand more about this experiment in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.